Hi everyone, welcome back to our channel, that is a study with entomologist. So this is the first video of the series where we are going to discuss the important MCQ topic wise. So in that the first topic is HPR, host plant resistant. So first question is use of resistant variety in IPM is the example of. So it is example of the cultural control. Next is earliest reported apple variety resistant to woolly aphid Eriosema lanigerum. So in our most of the book, the only variety that is winter mageddon is reported for the resistance against the apple woolly aphid, but in reality when the work was done at that time two variety was there. One was winter mageddon and the second one was the Siberian bittersweet. So these both are the resistant for apple woolly aphid, and Underhill is the variety which is resistant for the Haitian fly of wheat. Who saved French wine industries by grafting American rootstock with French scions of the grape? So the answer is the C.V. Riley. For that, C.V. Riley, for this work he got World Food Prize also in 1997. Next question is R.H. Pinter worked on. So R.H. Pinter worked on the Haitian fly. Next question is the first book on HPR was insect resistance in crop plant was written by. So it is written by R.H. Pinter. Species that are broadly adapted to fit successfully into a variety of habitat is called as. Already we discussed this. Uri means wide variety, and now he is asking about that variety of habits. So it's known as Uri topic, and if it is feeding on the wide variety of food habits, then the answer will be uriphagic, but here he asked the narrow. So steno means narrow, so the option will be stenophagic. Next question is major limitation in new variety development is. So for the new variety development, biotype can be easily developed. There are some genetical limitations also, and the conflicting traits can be grow in that particular time. So these all are the limitations for HPR can we say, and new variety development we can say. Next question is HPR is the relative amount of heritable qualities that influence the ultimate degree of damage done by the insect was identified. So this was identified Pinter. That's why Pinter is known as the father of HPR. Next question is optimal plant defense theory was given by. So this was given by the Cooley. So it is given by the Rhodes. Next question is strategies of insect to counter adapt plant defense. So sequestrations also, like he can store those toxic material, he can detoxify them, and gut bacteria mediated detoxification. So these all are the strategies to counter adapt plant defenses. Next is which of the following resistance can be easily broken by the insect? So we know monogenic, where the only one gene is working. So these types of resistance we can break easily. Next is which is the following is the field resistant. So we know horizontal resistance, which is minor resistance, and the polygenic resistance is known as the field resistance, and vertical resistance is known as the true resistance or the adapt resistance. Next question is it is race or biotype specific resistance. So vertical resistance, because it is only working against one or two. A very few genes are working on a particular thing. That's why it is a race or biotype specific. Next question is which of the following are not considered as true resistance? So pseudo resistance, apparent resistance, ecological resistance, these all are one only. That's why the answer will be the all. So answer will be all. Next question is pseudo resistance term was given by. So this term was given by Pinter. Pinter only give the name pseudo resistance. Next is type of pseudo resistance are host escape, host evasion, induced resistance. All three are the pseudo resistance not determined by the genetics of insects. Next question is a pseudo resistance type in which host may pass through most susceptible stage quickly. So it is known as host evasion. Host evasion. Next question is dual discrimination theory of HPR was proposed by, so it is proposed by Canada and Booth. How it can remember that here dual means two. Also the scientists are also two, Canada and Booth. Then nutrient imbalance theory was given by House in 1969. Next is botanical instinct theory was given by Bruce. B for botanical and B for Bruce also. Next question is chemical basis of host selection theory was proposed by, so it is proposed by Kogan, and Kogan and Outman gave the anti-zoonosis term also. The term non-preferences, antibiosis and tolerance was given by, so these are given by R.H. Painter. Next question is who is considered as father of HPR? So R.H. Painter is considered as father of HPR. Anti-zoonosis is also called as, so anti-zoonosis is also called as non-preference. The three important mechanisms of resistance like anti-zoonosis, antibiosis and tolerance was given by, so these are given by R.H. Painter. Next is this mechanism employed by the host plant to deter or drive away or reduce colonization in insect. So this is known as the anti-zoonosis. Only it has to deter. It don't have the adverse effect on the biology of insect, but just it will drive away from the plant. The next question is a compound released by an organism which evoke a response beneficial to number of another species but not to the another. So it is known as pheromones. Means for whoever it is taking, that is beneficial for them, not for the emitter. Next question is a compound released by one organism which evokes a reaction in an individual of a different species that is favorable to emitter but not the receiver. So it is known as allomone. Next question is oryzanone is attractant to, so it is attractant to striped stem borer. Next is repellent compound in sorghum against shoot fly, it is dhurin, that is HCN. Next is allylochemical gossypol is stimulant for ball weevil and deterrent to bollworm complex. So in that complex Helicoverpa is also there. That's why answer will be Helicoverpa. Otherwise you can keep the spotted bollworm, pink bollworm, spiny bollworm also. Next question is feeding stimulant for Bombyx mori. So it is morin present inside the mulberry plant, that is feeding stimulant. Next question is development of biotype is more in case of, so whenever there is a selection process, selection pressure against their life at that time, they will change more drastically inside them. That's why the development of biotype is mostly in the antibiosis cases. Breeders use dash as a main tool for development of resistant variety, so they use antibiosis as a main tool. Next question is dash prevent the biotype development as they do not have any selection pressure over the pest population. So these are known as tolerance, because those variety are giving some chance to feed on themselves, although they can tolerate the damage. That's why tolerance mostly not cause the biotype development or any kind of pressure on the insect. Next question is tolerant variety have a dash ETL than susceptible varieties. So tolerant varieties have higher ETL than susceptible varieties. Next question is tritrophic interactions one can find these all three: host plant, insect and the natural enemies. With these all the tritrophic interactions will be it. Then pheromone for trichogramma, so it is trichosan. This trichosan pheromone come from the scales of lepidopterans like Spodoptera and the Helicoverpa, through which this trichogramma get attracted. Next question is major pathway for the production of secondary metabolites like phenylalanine, tyrosine and the phenolic compounds in plants, and so it is through ischemic acid pathways. It is the important pathway which is used during the stressed conditions. Next is push-pull strategy was first described by, so it is described by the Pike et al. Next is maximum number of insect biotypes observed in Haitian fly, that is Mytiola destructor. These are 16. Next is maximum number of biotypes, insect biotypes are observed in which groups? So these are observed in aphids. Strategies to overcome biotype development: sequential release, horizontal resistance, gene pyramiding, gene staking. These all are the strategies for development of mechanism through which we can overcome the biotype. In the word, first insect transgenic plant was development in crop tobacco against the Manduca saxata tobacco hornworm. Next question is highest area of cultivation of transgenic plant is for the trait. So highest is present for the herbicide, followed by the insecticide, then herbicide and insecticide, and then virus. First transgenic crop in India. So in India the cotton was the first transgenic crop. And again we can say the first commercial transgenic crop in the world is cotton only. So in India the cotton came in 2002 with the Bolgard first generation. It's have the gene Cry1AB against the bollworm complex. Next is the largest area of BT transgenic crop at the global level is under. So it is under the first is soybean, then is maize. Here soybean is not in options, that's why maize will be the correct answer. Next is genetic engineering approval committee comes under the Ministry of, so it comes under the Ministry of Environment, Forest and Climate Change. Next question is Bolgard second contain. So Bolgard second contain two genes, Cry1AC and Cry2AB. Option B is the correct answer. And this generation released in India in 2006. Bolgard third is also scientists developed. It is made up of three genes, Cry1AC, Cry2AB and WIP3A, that is vegetative insecticidal protein 3A. Still it is not released in India, but Bolgard third is released in Australia. So remember these facts also regarding to the Bolgard. So these 50 questions for today, these are the from the